sharing that. So that was just a brief overview of some clinical aspects with regards to pneumococcal disease. Just as an introduction to our next talk, which is going to be hosted by Dr. Felix Dube. So Dr. Felix Dube, he is a UK Royal Society and AAS Future Leader of African Independent Research. Currently, he's a research fellow and senior lecturer at the Department of Molecular and Cell Biology at the University of Cape Town in South Africa. He obtained his PhD in medical microbiology in 2016, and currently his research focuses on the genomics and metagenomics for antimicrobial resistance surveillance. Also, very excitingly, Felix is a co-investigator on the REVIVE AMR sub-study. So Felix, I just want to say thank you very much for giving this talk today, and I will hand over to you, and then we will take some questions after Felix's talk.
Hi, Keisha. Good afternoon, everyone. Good morning to those who are on the western side of the world. Thank you so much for the kind introduction. And if it's all right, I'll just switch off my video because I'm working from two screens. What I would like to do today is provide some early tales from our longitudinal birth cohort, where we're looking at pneumococcal colonization dynamics in South Africa. Keisha, can you see my full screen?
No, we're not seeing your screen yet, Felix.
Is it up now?
Yes, it's up now.
Okay, great. Just want to... Okay. As Keisha has indicated, pneumococcus remains one of the leading bacterial causes of childhood pneumonia, with an enormous burden in terms of disease and death, especially in low middle income countries. We know that colonization by pneumococcus precedes the development of disease, and that the current vaccine, the pneumococcal conjugate vaccine that is being used, provides serotype-specific immunity, but this is insufficient, especially with the increase in emergence of serotype replacement disease. We also have an issue when it comes to an increase in terms of macrolide and cotamoxos resistance that we are seeing. But also we see that there are very limited studies or information relating to the impact of pneumococcal genetic determinants of transmission, acquisition rates, carriage duration, as well as antimicrobial resistance. So our study is essentially seeking to understand and investigate the colonization dynamics of pneumococcus in both health and in disease. So we, as indicated, there are many disease manifestations of pneumococcus, and this is in response to the pneumococcus seeking to colonize a diverse range of ecological niches. So in the first instance, what you get is that when you have local spread of the pneumococcus, you have disease such as pharyngitis, and if it migrates to the ears, you get otitis media, and in certain instances where you get microaspiration of the pneumococcus to the lower respiratory tract, you get pneumonia, and if you get a breach in the phagocytosis mechanisms, you get pneumococcus making its way into the bloodstream, causing septicemia, and if you have a breach in the blood-brain barrier, you get meningitis. We also appreciate that the pneumococcus, being a gram-positive bacteria, can be seen as success in terms of its its ability to inhabit different ecological niches, and that the proficiency of pneumococcus to actually cause disease makes it quite pliable in terms of driving the diversity and mutations that are seen and emergence of other serotypes that previously were not seen when the vaccine were introduced. We see that there are more than 100 different pneumococcal serotypes, and our current PCV13 has 13 serotypes indicated in red here. But what we are seeing is that the more we vaccinate with one vaccine, we get disease that is being caused by other serotypes. We see, for example, serotype 22F and 33F highlighted in blue, causing significant disease, pneumococcal disease, and hence leading to these two serotypes being included in the PCV15. And currently, I think GSK and Pfizer are looking at PCV20 and PCV40 as conjugate vaccines that can be used in children. When we talk about the pneumococcal capsule being the most immunogenic part of the bacteria, we are referring to the sugar coating that is on top of the bacteria. So if you look on the left-hand side, right on top of the bacterial cell wall, we see the capsule. And if you zoom in quite closely, it's these strands that in essence also cover some of these other antigenic peptides that protrude on top of the cell wall. So in essence, they get protected against being visible to host phagocytotic mechanisms. The diversity in terms of the capsule all starts with the capsular biosynthetic gene cluster, which is characterized by the differences in which the sugar residues are added continuously and translocated onto the cell wall. And based on the differences in these sugars that are added continuously, we get differences in the cell or in the bacterial capsule, and hence the different bacterial capsules that we are seeing or the serotypes that we are seeing. The question can be posed in terms of how do we then study and understand the carriage dynamics of pneumococcus? Well, we have nested our study in a longitudinal birth cohort that sought to determine the etiological risk factors for pneumonia, essentially looking at 7 risk factors that include genetic, microbiological, immunological, and other risk factors. My interest and my group's interest is really on the microbiological side, where we're looking at the roles that bacteria, viruses, and fungi play in driving the progression from just colonization to actually causing disease. In this case, the disease outcome that we're looking at is pneumonia or lower respiratory tract infection. So when you look at these environmental exposures that could include different bacterial species, smoking, feeding practices, vaccination, which provides a selection pressure, antibiotic use, these play a role in terms of perturbing or disrupting the healthy microbial profiles of the nasopharynx, which is the ecological niche of pneumococcus. So if we get direct extension or invasion of these of the epithelial linings, either in the nasopharynx or in the lower respiratory tract, we get disease manifestations that include pneumonia or meningitis. But we also know that these diseases can also result into the remodeling of the lungs, resulting in things such as asthma. We can also have skewed reprogramming of the immune system that from colonization can also drive the progression of asthma. So we have the exposures, we have the microbiome, and we have the outcomes. So in terms of the outcomes here, we're looking at the colonization dynamics of pneumococcus. And to study this, as I mentioned, we have nested this into a longitudinal birth cohort where we enroll children from birth and follow them every 2 weeks for the first year of life, right? Up to 12 months there. up to 12 months, and also at any other scheduled clinical visits, and 6 months after the first year of life. Most importantly, this study is located in the Drakenstein area, which has a very distinct population that comprises of admixed ancestry in Tisi Newman and Mbekweni, which is largely a Black African population. And I'll come back to this in terms of how this plays a role in terms of the diversity of pneumococcal lineages that we are seeing in our cohorts. So once we enroll the children into the study, we collect nasopharyngeal samples. And in this context, we have collected more than 29,000 nasopharyngeal swabs that were cultured on 2% blood agar and AMR tested nucleic acid extracted from the resulting bacterial colonies that we are growing on the 2% blood agar. And after that, we have then moved on in terms of confirming whether indeed what we are seeing on the plate is pneumococcus. And if it is pneumococcus, we then go on to sequence this and determine different aspects and understand the genome of the pneumococcus in relation to the child in whom we have isolated this pneumococcus. So the outcome here, we are looking at the colonization by pneumococcus, but we also know that the child within this first year of life, that they can have repeated episodes where they can be recolonized by a non-vaccine serotypes, or in essence, lose this non-vaccine serotype and acquire a vaccine serotype, or have periods where they are not colonized by any pneumococcus in the green state here. So we use Markov multi-state modeling because of the different states in which we find these children, to kind of understand how the carriage dynamics change over the first year of life and how that relates to disease. And when we're talking about disease, there are two approaches that we can use. The first approach is where we have a child at the point of disease, and because of the longitudinal nature of this study, we can go back into the periods preceding the development of disease and compare the lineages at the time of pneumonia and compared to the times before the development of pneumonia. Another strategy is just to look at all the isolates and compare isolates that were collected in the non-pneumonia episodes to those that were collected at the time of pneumonia. And this provides us with better insights into the colonization dynamics of pneumococcus in both health and in disease. So when we want to look at the effect of host co-infection and strain-specific factors in driving pneumococcal carriage acquisition and duration, the things that we're interested in at an individual level, we're looking at the age, we're looking at the vaccination status, antibiotic exposure, we look at household levels, as Keisha indicated, that overcrowding is a risk factor for pneumococcal acquisition. We also look at the maternal risk factors. We look at other microbes that could be isolated at the time that we detected a pneumococcus, and then we look at the bacterial or strain level factors that may drive the acquisition and carriage durations of pneumococcus. As I indicated, that we study the colonization dynamics by using a Markov multi-state model, where we can have repeated episodes where a child is not carrying a pneumococcus, and in certain instances where the child acquires a pneumococcus, that they can be persistently colonized by pneumococcus. And we want to understand what are the factors that actually drive persistent colonization by the same or different pneumococcal serotypes over the first year of life. So in terms of the maternal characteristics that we are seeing, most importantly, we see that in our cohort, we have high prevalence of smoking, HIV exposure, as well as TB contacts. When we look at the infant level characteristics, we see that children that are born from Mbekweni have a high HIV prevalence compared to those that are born from mothers in TC Newman, which is an admixed ancestry, right? And the converse is true when you look at birth weight as well as smoking exposure, and this is based on cotinine levels. So when you look at the first year of life, right, from birth up to 1 year, we're seeing that the carriage prevalence of pneumococcus gradually increases, such that by 14 weeks, more than 50% of the children were colonized by pneumococcus. We see also that we have been successful in terms of culturing pneumococcus, in terms of having cultured more than 76% of samples being positive for pneumococcus. And of these, we have been successful in terms of sequencing these isolates in the black bars, right? We have similar numbers of samples that were sequenced from TC Newman compared to Mbekweni, hence bias being restricted or minimized. When we compare the coverage or the proportion of vaccine serotypes in red versus vaccine serotypes in blue, when I say vaccine serotypes, I'm referring to the 13-valent pneumococcal conjugate vaccine, which includes 13 serotypes. We see that we have a significant proportion of pneumococcus that are non-vaccine serotypes, and about 20% throughout the first year of life being PCV13 serotypes. When we fragment this to at a more granular level, we see that there are certain serotypes that are quite interesting. If you look at the yellow bar here, which is serotype 19F, regardless of the vaccinations that are given at 6, 14, and 9 months, we still see similar proportions of serotype, residual coverage of serotype F. I also want to draw your attention to serotype 16F, which are just the two serotypes that I've cherry-picked for further discussions in the slides that follow. And we also see that with each subsequent dose of PCV that is given, we get a reduction in the proportion of serotypes that are carried after the first, second, and third dose of the vaccine. And if we come back and look at the sites themselves in relation to the serotypes that are being carried, we see for vaccine serotypes that serotype 19F, for example, is the most common vaccine serotype that we are seeing, slightly different between sites. And if we also look at the non-vaccine serotypes, we see that serotype 16F is also one of the predominant non-vaccine serotypes. If we contextualize this data in terms of resistance to antibiotic classes, we see that 19F, for example, is resistant to more than 7 different classes of antibiotics, which has significant public health relevance, right? And then we also see non-typeable pneumococci also being resistant, multi-drug resistant, and serotype 15A, some level of resistance we are seeing also in serotype 16F. I'll come back to these two serotypes as we proceed. In terms of the effect of strain level or household level factors in pneumococcal carriage, we see that daily carriage probabilities are higher between pneumococcus and Haemophilus and Moraxella catarrhalis, which is consistent with what is being reported in the data and data from the Gambia. We also see this when we subsample our cohort in terms of looking at those children that develop disease to those that do not develop disease. In this context, we're looking at pneumonia. We're seeing that pneumococcus is positively correlated with Haemophilus influenza, Moraxella. We also see that there's an increase in the number of the probability of acquisition is higher in children where the number of children in a household is higher. So here we're referring to overcrowding, which is one of the risk factors that Keisha highlighted in her earlier presentation. We also see that lower children that are from lower socioeconomic status have also lower clearance probability. So in this case, we're referring to longer carriage duration. And with this, it provides an opportunity for pneumococcus, which is naturally recombinogenic, to undergo homologous recombination and allow foreign DNA to come in. And this is important when we talk about capsular replacement, and especially in the context of replacement disease that we are seeing. We come back now that we've looked at just a general overview of the carriage dynamics of pneumococcus, to actually seeing the genetic determinants of disease progression using a GWAS model. And our rationale here is that the unmeasured contribution of pneumococci in non-bacteremic pneumonia will manifest as differences in pneumococcal strains between children that develop pneumonia to those that do not. So remember, initially I mentioned that we can do this in two ways. We can look at the child at the point at which they develop pneumonia, right? And look at the preceding time points and compare the strain level or serotype level differences between the cases in red and the controls in green. And if we follow this approach and we look at modeled prevalence of pneumococcus, we see that in the 90 days preceding the development of pneumonia, that the prevalence of pneumococci in controls to those with non-severe pneumonia is similar, right? But the prevalence of pneumococcus in severe pneumonia cases is lower compared to these two. But in about 30 days before the development of pneumonia, which is at 10.0, we see a drastic increase in the prevalence of pneumococci in children that went on to develop severe pneumonia. And we see this when we and look at specific serotypes such as serotype 23F, we can see that in the 60 days prior to the development of pneumonia, that there's a gradual increase in terms of the prevalence of serotype 23F in the periods preceding the development of pneumonia. And the is true when we look at serotype 10A, which is a non-vaccine serotype, perhaps suggesting that being colonized by serotype 10A might be protective against development of pneumonia. We can also look at risk factors which are similar to what we looked at from the overall study cohort in terms of the birth weight, HIV exposure, as well as season at which the child was born. The other thing that we can look at is to look at the maximum likelihood phylogenetic tree, in which we can contextualize the genetic data of the genomes that we have sequenced with metadata that we have collected. So we see, I'll just highlight one or two serotypes, right? So just to orient ourselves, the inner ring refers to the different serotypes which are listed here. And then as we go from the inner ring, the first one, as we go outwardly, we are looking at the different antibiotic resistance. This is in silico resistance to a number of antibiotics, starting with penicillin, which is the red, going outwardly to blue, which is another antibiotics, and then the yellow represents susceptible isolates. So wherever we see yellow, it means that the strains or the isolates that were collected are susceptible to that antibiotic or to the different antibiotics. So when we look at non-typable pneumococcus, we see that most of them are multi-drug resistant, which is consistent with the literature. I also highlighted serotype 19F, which was resistant to more than 7 classes of antibiotic. And when we look at the genetic data, we can actually see resistance to not only penicillin, but to other macrolides, as well as cotramoxazole. We see multi-drug resistance again in other serotypes, such as 15A, and also highlighting your attention to serotype 16F, which is largely resistant to cotramoxazole. I'll come back to this in a moment. The outer ring shows the disease state, with the green being carriage isolates and the yellow being non-severe pneumonia. And we can see that the majority of the isolates were collected from carriage as opposed to disease. We can also look at this by looking at the pneumococcal, the global pneumococcal sequence clusters, which is a typing technique that is more superior from the conventional MLST profiles that only look at 7 housekeeping genes, whereas the GPSC model looks at KMAs, which has a higher resolution compared to MLST profiles. So at the bottom here, we're seeing carrier status, non-severe pneumonia or pneumonia, and the striped bars represent non-vaccine serotypes. I've tried to indicate which serotype it is. So we have a lot of 16F that are seen in carriage compared to disease. In certain instances, such as 15B, we don't see differences between the carrier state and the disease state. We see for GPSC 46, which is one of the lineages under 16F, being resistant to cotramoxazole, which is consistent with what we are seeing in terms of resistance to cotramoxazole that we saw in the phylogenetic tree that we looked at. For serotype 19F, we see that GPSC5 is resistant to both cotramoxazole as well as macrolides and penicillin, right? And similar things can be seen for the other GPSCs. We look at the differences in the serotype distribution in children presenting with pneumonia, and we see that children with pneumonia are more likely or often colonized by serotype 19F compared to the controls. The converse is true for serotype 13. And these are the differences in terms of antibiotic resistance in children with pneumonia compared to those without pneumonia. And when we come to the granular level of the GWAS analysis, that is we see that there are certain genes that are more common in children that presented with pneumonia. Specifically, if we look at FPUC, which is an ATP binding cassette transporter that is important in terms of acquisition of Petrobacter, which is a siderophore that is important in terms of ion sequestration. So we know that one of the host defense mechanism against bacterial infection is to sequester ion from the bacteria. And we see this role being played by a lot of macrophages. And in response, some bacteria produce these siderophores that help in terms of compete for this ion, which is important for a number of metabolic processes in these bacterial species. And we see an upregulation of this gene in children that develop pneumonia. And there are a number of other genes that we're seeing to be associated with disease. I won't spend a lot of time on this, but what I want to do now is to perhaps cherry pick 2 serotypes. One, which is serotype 19F, which is a vaccine serotype, and the other being serotype 16F, which is a non-vaccine serotype, and kind of understand the population structure of these strains, right? In context of their ability to progress from just being normal colonizers to actually going on to cause disease. We contextualize this data in terms of the national data that looks at the invasive disease that is caused by serotype 19F. So this is data that has been collected in South Africa by the NICD and includes data looking at different pneumococcal serotypes. And in this case, we've just narrowed it down to serotype 19F and looking at the different lineages within serotype 19F. And we see that after the introduction of PCV7, so PCV7 has 7 serotypes, so it's a 7-valent vaccine. We see a drastic reduction in GPSC1 and further reduced after the introduction of PCV13, which is the 13-valent pneumococcal conjugate vaccine. So this is in disease, right? And it's closely mirrored by data from COURAGE studies, both from Soweto in Agincourt as well as in Cape Town. In our context from the Drakensberg area, except that we see that in Soweto, GPSC 21 was the most predominant serotype 19F that was seen post introduction of PCV 13. Whereas if we look at the Drakensberg area, in the duration of our study, we see a gradual switch between GPSC 1 to GPSC 205. And I'll come back to why this is important and why we continuously then seeing residual coverage of 19F despite a high vaccine coverage, which is in excess of 98%, right? And partly that can be explained by these lineages that we are seeing emerging post-introduction of PCV 13. When we look quite closely at the capsular biosynthetic gene cluster, right, what we are seeing is that there's a variant of serotype 19F GPSC1 that has GLF1 and GLF2 that we don't see in other lineages, as you can see from the top. We also see a number of these being included in GPSC1. And we'll come to understand the importance of this when we look at the different variants. We've looked at this data from a global context, and we're seeing that much of these serotypes are detected in South Africa, as well as some parts of Asia and some parts of Europe. But most importantly, what we are seeing is that those that are GLF positive 19F, these are more likely to cause disease compared to COURAGE. So we end up with two variants of serotype 19F GPSC1, which include variant 1, which has the GLF1 and GLF2, whereas variant 2 has insertion sequences in the same area within the genome where we're seeing GLF1 and GLF2. And when we look quite closely then, so to what extent do these genetic differences translate into how the capsule interacts with serum or phagocytosis that we are seeing? What we see is that when we compare the ability to phagocytotic killing by serum that was obtained from adults that were immunized with PPV23, that is the pneumococcal polysaccharide vaccine. This is not conjugated to a peptide, right? So it's just the polysaccharide that is included, that is derived from 23 different serotypes. What we are seeing is that GPSC1 that is associated with clonal complex 420 and GPSC 205 are more likely to be killed by serum that is obtained from these adults compared to GP1 from clonal complex 320 and GPSC 21, which are less likely to be killed by serum from adults immunized with PP23. We see this when we also look at serum that is obtained from children that were vaccinated with PCV13, that when we look at GPSC1, consistent with what we are seeing from serum killing from PPS23, as well as PCV13. So in essence, what we are seeing, we have a higher percentage of killing that is being observed by serum from children immunized with PCV13 as compared to adult serum from adults immunized with PPV23. And this is consistent with the literature, right? We get better immune programming when we conjugate the polysaccharide to an antigen that solicits the help of T helper cells that is able to then reprogram the immune maturation and hence better immune response to pneumococcal challenge. When we look at serotype 16F, what we're seeing is that what we've done is that we have collected from the 1,137 children, we've ended up with a subset of 10,000 genomes from which we have subsampled and looked at the distribution of serotype 16F. As we saw previously, serotype 16F is one of the predominant non-vaccine serotypes that we are seeing in our cohort. And the major lineages that we see in serotype 16F are GPSC 33, GPSC 46. And we see that these are quite distinct in terms of the antimicrobial resistance profiles. We're seeing that with GPSC 33, it's resistant to both, especially from South Africa, that they are resistant to both penicillin as well as cotramoxazole, right? And then if we look at GPSC 46, we're seeing that it's also quite resistant to cotramoxazole and partially and very little resistance to penicillin seen in this lineage. And how does this translate in terms of disease? We're seeing that GPSC 33 is much older compared to, in terms of how it emerged from the most common ancestral lineage that we're seeing compared to GPSC 46. Sorry, I'm just rushing this through because of time. I'm happy to take any questions after the talk. This highlights the importance of this serotype and perhaps helps us pose questions around the possibility of emergence of serotype 16F as one of the replacement serotype. And recent data is increasingly showing that 16F is an emerging pathogen that is causing lower respiratory tract infection in many settings. So to conclude, we're seeing that despite a very high and excellent immunization program in our context, we still see residual coverage by vaccine serotype. We also see distinct lineages that are seen in disease compared to health status. We also see that pneumococcal strains of the same serotype, for example, when we looked at serotype 19F, exhibit differences in terms of invasiveness as well as resistance to serum killing, either by serum obtained from PPV23 or PCV13. We also see that the higher resistance to serum killing in GPSC1 and 21 may explain, therefore, the persistence of this lineage in terms of courage as well as in IPD. This argues for continued surveillance of pneumococcal courage in settings where we have high courage of pneumococcus as it relates to disease in children. I would like to thank participants in our study, as well as the members of the Dube Lab that have played a significant role in terms of making this work possible. Thank you so much.
Thank you very much, Felix. Thanks so much for that presentation and for that talk. I know you had a lot to get through in very little time, so thank you very much for that. I'm sure we all learned something from your talk today. We are just on the hour, so we have run out of time. But Elsa has put in to the chat any if anyone has any questions to direct to Felix, they're more than welcome to. And Graham,
yeah, thanks, Kisha. Is there time for one question?
I think we can. I think we can.
Great.
Thanks,
thanks, Felix and Kisha for a great session. Highlighting the biology and the molecular epidemiology of pneumococcus and the clinical aspects. I think it was a great session, and we don't spend enough time discussing this very important pathogen in people with advanced HIV disease. Felix, I've just got one general question. if you can just address the issue of the effect of antibiotic exposure on pneumococcal carriage, and specifically azithromycin, if you've done any work on that.
Thanks, Graham. So we are yet to do work in terms of looking at azithromycin, especially in the Drakenstein. So when we look at the Drakenstein cohort, we have very little exposure to azithromycin, and compared to other studies that we've conducted, such as the chronic lung disease cohort, where we expose children to weekly doses of azithromycin. And what we are seeing is that as we increase the selection pressure of azithromycin on the bacterial population, we get increase in terms of antibiotic exposure. And when we cease and have a wash-off period, we kind of have the antibiotic resistance levels reverting to what we're seeing at baseline, right? But what is interesting is that there are other closely related species of Streptococcus that end up harboring antibiotic resistance genes through mobile genetic elements that they can in turn also transmit to pneumococcus, and hence creating a pool of potential sources of antibiotic resistance genes. So for a study such as the REVIVE trial, it's going to be very important to look at this longitudinal impact of increased antibiotic pressure and characterize the mechanisms in which antibiotic resistance develops.
Right. Thanks, Felix.
Great, thanks for the question, Graham. Thanks, Felix. All right, we are out of time. I see Tafesa has put a question in the chat. I will we'll respond to that via email. But thank you very much to everyone for joining us today. I think we had a great presentation by Felix, and we're very excited to see the work come out of the AMR sub-study as well for REVIVE. So please look out for our next seminar, which will be in approximately a month's time. And we look forward to seeing everyone there. And please feel free to share that invite as well. So thank you very much, everyone, for joining today.
Thanks.
Thanks.